Yes, this would be the second module for the finals, your module 8, which will tackle about the immune regulation. Now, when you say immune regulation, how the immune system is being controlled. So, we will tackle the different cytokines that would regulate the immune system. So, let's begin. So here, class, in regulation, the one involved here are your cytokines. Now, as you all know, cytokines, class, are small, soluble proteins that would regulate the immune system, orchestrating both innate immunity and the adaptive response to infection. They also serve as chemical messengers produced by several different types of cell, have activity modulating effects on both hematopoietic and immune systems through the activation of cell bound receptor proteins. Now, cytokines are induced in response to stimuli such as the presence of lipopolysaccharide or flagellin, through ligation of cell adhesion molecules or through recognition of foreign antigens by host lymphocyte. If you still remember, class, di ba, ang cytokines nyo, they are produced in the presence of antigens such as the ones that contain lipopolysaccharide and flagellin. So, itong mga to, these are your bacteria. Bacteria would have lipopolysaccharide cell wall and they would have flagellin. Now, when this happens, they would ligate or they would attach to certain adhesion molecules. Now, they would direct also the growth, differentiation, and gene expression by many different cell types, including your leukocytes. Now, this is the mechanism, class, on how your cytokines would be produced. Cytokines would act only on cells bearing specific receptors. So they have a specific target class that have specific receptor. And expression of cytokines and the receptor is highly regulated. Please remember that they are highly regulated. So whenever there would be an inducing stimulus, when you say inducing stimulus, let's say nagkaroon ng lipopolysaccharide or nagkaroon ng flagellin, certain cytokine producing cells would cause their cytokine gene to become activated. So let me repeat. When your cytokine producing cell encounters a stimulus like your lipopolysaccharide, flagellin, or any presence of antigen molecules, this will cause the cytokine gene to be activated and the cytokine producing cell will start producing cytokines. And as mentioned earlier, itong cytokines nyo, class, will only act on cells with specific receptors. So the cytokines, class, will go to target cell. Pupunta sila dun sa target cell nila with their target receptor. They would attach. They would attach and it would send a signal causing the target cell for its gene to be activated, leading to different biological effects. So let me repeat for the third time, class. When your cytokine producing cell encounters a stimulus such as your lipopolysaccharide, your flagellin, or any antigen molecules, it would cause the cytokine gene to be activated, producing cytokines. And as mentioned, cytokines will go to their target receptors on the target cell. Once they are in the receptor, they will send a signal causing the activation of your target cell, causing different biological effects, which we will tackle in the next slides. Now, there are three modes of cytokine action, class. Please remember this. The first one is your autocrine. When you say auto, again, this would refer to yourself. So when you say autocrine, this is same cell would secrete and receive the cytokine signal. Sa mas madaling salita, here, class, the cytokine producing cell and the target cell are the same. Let me repeat, in autocrine class, the cytokine producing cell and the target cell are the same. So same cell would secrete the cytokine and would also be the one to receive the cytokine signal. So pagka-release niya class, pagka-release niya ng cytokine, lalabas yung cytokine, it would also be received by the receptor of the same cell. So I hope that is clear. The second type is your paracrine class. Paracrine class, please take note, is that this is a cytokine signal secreted to a nearby cell class. Nearby cell. So here you have your cytokine producing cell or your secreting cell. The cytokines will go to a nearby responding cell. So pakitandaan lang class yung keyword natin. Pag paracrine, nearby cell, malapit lang. Kapag naman endocrine, the last mode of cytokine, cytokine signal secreted to the circulatory system. So if there is a circulatory system class, there is what we call travel. Your cytokine will travel class. And the target are distant cells. So let me repeat, if it's endocrine, there is the involvement of the circulatory system wherein your cytokine will travel to target distant cells. So this is your CPC or your cytokine producing cell. It will secrete the cytokines. It will go to the blood vessel and it would go to the target cell that is distant responding cell. So just a recap, pag autocrine, self cells. Cytokine producing and target cell are the same. Pag paracrine, nearby yung target cell. Kapag endocrine, distant yung target cell. Now, cytokines class are pleiotropic, meaning class they have pleo from the prefix pleo meaning many. Cytokines class can have different effects on different cells. So when you say pleiotropic, one cytokine can have different effects on different cell. Example class. Here is your cytokine producing cell. The cytokine producing cell here is your T helper. Now, si T helper class will secrete the cytokine. It will secrete the cytokine interleukin 4. Now, si interleukin 4 class, for every cell type that it would target, it would have different biological effect. Let me repeat class. In this picture, the cytokine producing cell is your T helper cell or your activated T helper. It will secrete the cytokine interleukin 4. And interleukin 4 class, which is the cytokine, will have different effects on different cell. This is the biological effects na class that I mentioned earlier. So, interleukin 4 on B cell will cause activation, proliferation, and differentiation. So, interleukin 4 class on B cell can cause its activation. It can proliferate, it can differentiate into a memory cell or a plasma cell. Interleukin 4 on thymocyte will cause proliferation, dadami sila. Interleukin 4 on mast cell will also cause proliferation. So I hope that is clear on what pleiotropic means. One cytokine can have different effects on different cells. Now, cytokines class can also be redundant. When you say redundant, sa Tagalog, paulit-ulit, repetitive. These are cytokines that can have the same effects. Example again, class. Here is your cytokine producing cell or your T helper cell. It will secrete three types of cytokines: interleukin 2, 4, and 5. Now, all of these three class has the same effect on one target cell. So, interleukin 2, 4, 5 on B cells will cause proliferation. So, I hope that was clear kung ano yung redundant. So, different cytokines can have the same effect on a single target cell. Now, cytokines class can also synergize with each other. When you say synergize class, they can work together or they can fuse. Now, example, again, activated T helper is your CPC or your cytokine producing cell. It will secrete interleukin 4 and 5, and together, when they act together class on the B cell, it would cause your B cell to induce class switch to IgE. So please take note of that. Then they could also antagonize one another. When you say antagonize, they would counter one another. An example of this, let's say, ito ulit yung cytokine producing cell nyo. Activated T helper will produce interleukin 4 and interferon gamma. Now here class, interferon gamma would counter interleukin 4. It would counter interleukin 4 causing the blockage or the cessation or the stoppage, stop niya. It would stop interleukin 4 from inducing a class switch to Ig. So that's your antagonize. Now there is a so-called cascade effect class. Now ano ba tong cascade effect na to? In a cascade effect class, cytokines can stimulate the production of other cytokines. So an example of this is your CPC. It would secrete the cytokine interferon gamma. Now, si cytokine interferon gamma would have the target cell macrophage. Now, when this happens, when your interferon gamma cytokine targets your macrophage, it causes your macrophage to now become a cytokine producing cell, releasing now the cytokine interleukin 12. And interleukin 12 would have the target cell activated T helper, causing activated T helper also to secrete interferon gamma, tumor necrosis factor, interleukin 2, and other cytokines. So that is your cascade effect class. Para siyang domino. The so-called domino effect, wherein one cytokine producing cell will cause the activation of another cell to produce cytokines. So that's the cascade effect, or for me, it's a domino effect. Now, classification of cytokines. The first one is their structural groupings. The first one is your interferons. The second is your tumor necrosis factor. The third one is your interleukins 1 to 32, your transforming growth factor, and your colony stimulating factor. So you're already familiar class dito, itong dalawa na to. This is included in your hematology. If you still remember in your hematopoiesis, dinidiscuss yan doon. But we will tackle for some of the cytokines na involved dyan, specifically on immune regulation. Now, another one would be functional grouping. So yung first one is structural. The second group is functional. So the first functional group is the mediators and regulators of the innate immunity. So these are the cytokines that would act on endothelial cell and leukocytes to stimulate early innate response. Then we also have mediators and regulators of the adaptive immunity. So these are cytokines naman class that would act on lymphocyte to stimulate and regulate adaptive response to specific things. Then we also have stimulators of hematopoiesis. They would act on the bone marrow to stimulate growth and differentiation of leukocytes and your lymphocytes. Now let's tackle the first group class, the cytokines of the innate immune system. So here we have your type 1 interferon, interferon alpha, and interferon beta, interleukin 1, 6, 12, 15, 18, tumor necrosis factor alpha, TGFB, or your transforming growth factor beta, and your chemokines. So let's begin. Let's start with the first one, your type 1 interferon. So this would include interferon alpha and interferon beta. Now they are responsible for interfering with viral replication and cell division class, meaning they are antagonistic. They counter they counter certain cytokines. They interfere with viral replication and cell division. They activate natural killer cells and would enhance class 1 MHC expression, thus increasing recognition and killing of virus infected cell. The origin class, the one that would secrete the cytokine producing cell for interferon alpha is your leukocyte, while the source or the CPC naman for interferon beta is your fibroblasts and epithelial cell. Now let's go to your second one, interleukin 1. Now interleukin 1 class would consist of interleukin 1 beta, interleukin 1A, and interleukin 1RA. So tatlo sila class under interleukin 1. Interleukin 1 beta, interleukin 1A, and interleukin 1RA. The cytokine producing cell here, or the one that secretes it, is your monocyte and macrophage. So this would act as an endogenous pyrogen class, please remember, and induces fever in acute phase response through its action in the hypothalamus. Now, interleukin 1 would also induce the production of vascular cell adhesion molecule as well as chemokines and interleukin 6 class. Please remember this, it creates or produces interleukin 6. Now, under interleukin 1 is your interleukin 1RA. This is an antagonist class to interleukin 1 and it blocks the interleukin 1 receptor, limiting the availability of the receptor for interleukin 1. So let me repeat. Si interleukin 1RA no class is an antagonist to interleukin 1. It blocks the receptor, limiting its availability. This in turn would help to regulate the physiological response to interleukin 1 and turn off the response when no longer needed. Then the third one, interleukin 6, the source or the cytokine producing cell is the macrophages, the T cells, and your osteoblasts. This is a part of the cytokine cascade release in response to lipopolysaccharide and would play an important role in acute phase reaction and in the adaptive immune response. So part daw siya class ng cytokine cascade. As mentioned earlier, di ba nga yung interleukin 1 nyo? It can cause the secretion of interleukin 6, which is again a part of a cascade effect wherein one interleukin, when one cytokine would cause the creation of another cytokine. It plays a role in acute phase reactions and adaptive immune response. Now, interleukin 6 is expressed in a variety of normal and transformed cells, including your T cells, your B cells, your monocyte, macrophages, fibroblasts, hepatocyte, keratinocyte, astrocytes, vascular endothelial cell, and various tumor cells. So aside from normal cells class, si interleukin 6 nyo could also be found in transformed or abnormal cells. Now, interleukin 6 would stimulate B cells to proliferate and differentiate into plasma cells and would induce CD4 positive T cells to produce greater quantities of both pro and anti-inflammatory cytokines. Then let's go to the fourth one, your interleukin 12. Ano ba tong interleukin 12 yun na to? This is the source is the B cell and the macrophage. It stimulates the production of interferon gamma by natural killer and T cells, stimulate the differentiation of naive T cells into T helper 1 cells, and would enhance cytolytic functions of activated NK cell and CD8 positive T cell. Growth factor for activated NK LUC cells. So please remember this. This is the growth factor class for activated NK LUC cell, your interleukin 12, also known as the NK stimulatory factor. Then we have your interleukin 15 class. The CPC or the cytokine producing cell is the T cell, produced in response to viral infection and other signals that would trigger the innate immunity. Promotes proliferation of NK cell, produces survival signals for memory lymphocytes. It synergizes in LUC cell induction process. It increases anti-tumoral activity of T killer and NK cell and can be chemoattractant for T lymphocyte. Endogenous interleukin 15 is a key condition for interferon gamma synthesis. Please remember this. It is required class for interferon gamma synthesis. Then we have your interleukin 18. Origin class is your macrophages. Acts as a synergist with interleukin 12, which stimulates the production of interferon gamma by NK and T cell. High interferon gamma production under integrated effect of interleukin 18 and 12 would suppress tumor growth. So when you have interleukin 18 plus interleukin 12 class, this will cause the production of your interferon gamma, which can lead to decrease tumor growth. So one requirement for the creation of your interferon gamma is the combination of your interleukin 18 and interleukin 12, which is important for decreasing or suppressing tumor growth. Then we have your tumor necrosis factor alpha, also known as your cacictin. Your cacictin class is produced by macrophages. It activates T cells to its ability to induce expression of MHC class 2 molecules, vascular adhesion molecules, and chemokines. Now, when selected at higher levels, TNF can have deleterious systemic effects, leading to septic shock, causing a decrease in blood pressure or hypotension, reduced tissue perfusion, and disseminated intravascular coagulation. The latter may lead to uncontrolled bleeding. Then we have your transforming growth factor beta or your TGF beta. The origin here class is your T cell. Now, active transforming growth factor is a regulator of cell growth class, differentiation, apoptosis, migration, and the inflammatory response. Thus, please take note that it is a control to help down-regulate the inflammatory response when no longer needed. it is considered as an inhibitory cytokine. Now, transforming growth factor beta would regulate the expression of CD8 in CD4, CD8, CD4 negative, CD8 negative thymocytes and would act as an autocrine inhibitory factor for immature thymocytes. So siya yung nagre-regulate class sa mga double negative na mga thymocytes. It inhibits the activation of macrophages and the growth of many different somatic cell types. And it is also an anti-inflammatory factor for mature T cell. Transforming growth factor beta blocks the production of interleukin 12 and strongly inhibits the induction of interferon gamma. Please take note of that. Now let's go to another group of cytokines this time class, yung tinatawag nating chemokines, which is also part of the innate immune system. So a chemokines class is a family of cytokines that enhance motility, promoting migration of many types of WBC toward the source of the chemokine. The target cells are usually macrophages. They have different groupings. So, na ano na natin to in the previous module ng midterms class. Yung C chemokines, CC chemokines, CXC chemokines, and CX3C chemokines. Now, functional grouping. Chemokines class are grouped into their function. They could be homeostatic. When you say homeostatic, this is the one that involves the homeostasis. So this is very important class for normal development or normal growth. So this controls the migration of cells during normal development and maintenance of tissues and lymphoid organ. Now another grouping, functional grouping ng chemokines nyo is they are considered inflammatory chemokines. Produced in response to infection or injury, and they would direct the migration of leukocytes into the infected or damaged site. Then we have your angiogenic chemokines. These are chemokines that will promote the development of blood vessels. They are pro-angiogenic. Others prevent the development of blood vessels. They could either be also be anti-angiogenic. Now let's go to the second part, your cytokines of the adaptive immune system. This would include your interferon gamma and interleukin 2, which are T helper 1 cytokines. Then interleukin 4 and interleukin 10, which are T helper 2 cytokines. There is also, let's continue. So let's start with your interferon gamma class. So the CPC or the cytokine producing cell here or its origin is your T helper 1. Now this is responsible for regulation and activation of CD4 T helper 1 cells, CD8 cytotoxic lymphocytes, NK cells, bactericidal activities, interleukin 12R and interleukin 18R. So please take note of its function class. Now, interferon gamma would stimulate antigen presentation by MHC1 and MHC2 molecules, stimulator of macrophages, and would boost their tumoricidal activity. Then let's go to your interleukin 2. This is also known as your T cell growth factor. Its origin is the T helper cells. It drives the growth and differentiation of both T and B cells and would induce lytic activity in NK cell. Interleukin 2 and interferon would induce the development of T helper 1 cells. Please remember this class, interleukin 2 and your interferon would induce the development of your T helper 1, which in turn would induce macrophage activation and delayed type hypersensitivity. Now, interleukin 2 alone can activate proliferation of the T helper 2 cells and would help to generate IgG1 and IgE producing cells. Then let's go to your interleukin 4. The origin is T cells and mast cells. They induce the differentiation of naive helper T cell or your THO. This is the inactive form class ng helper T cell nyo to become your T helper 2 cell. So, ang interleukin 4 nyo is responsible for turning the inactive T helper to T helper 2. It promotes the production of IgG2A and IgE along with interleukin 5, drives the differentiation and activation of eosinophils in both allergic immune responses and response to parasitic infection. Naalala nyo pa, di ba, si T helper 2 nyo class is involved in allergic and parasitic infections. So, tumutulong din ang interleukin 4 nyo siya dyan sa allergic immune response and parasitic infection. Then we have your interleukin 10. So, the origin would include monocytes, macrophages, CD8 positive T cell, excuse me, and T helper 2, CD4 positive T cell. Now, its function is to inhibit activated macrophages, displays potent abilities to suppress antigen presenting of APC. Released by T cytotoxic cells to inhibit the action of NK cells during immune response to viral infection. It is predominantly class an inhibitory cytokine. So remember that this is the inhibitory cytokine of your adaptive immune system, si interleukin 10. Then we have your interleukin 5. The origin is T helper cells and your mast cells. It activates eosinophils and would serve as link between T cell activation and eosinophilic inflammation. It would stimulate growth and differentiation of eosinophils and activates mature eosinophils. Growth and differentiation inducing factor for activated T and B cells would induce class specific B cell differentiation involved in IgA production class is to be specific. So siya yung tumutulong sa pag specify to become an IgA antibody, si interleukin 5. Then we have your interleukin 13. The origin is your T cells. Now, this is similar to interleukin 4 but appears to have less effect on T or B cells than interleukin 4. Inhibits activation of macrophage and would counter or antagonize interferon gamma. Please remember this. They antagonize or they counter interferon gamma. Important mediator of allergic inflammation and disease. Now, these are the different cytokines of hematopoiesis. Kindly take note of them, class. They would be involved in your exam. This will not come in your quiz. Hindi siya lalabas sa quiz, but it will come out in your finals. So, please try to memorize them. Now, I want you to take note here, class, of the G CSF, the M CSF, and the GM CSF. CSF here would stand for the colony stimulating factor. Now, they are imperative or very important class in your hematopoiesis. So, do take note of that. Now, yung G dito, this would stand for granulocyte. Yung M dyan would stand for the macrophage. So, GM would stand for granulocyte macrophage colony stimulating factor. So, they have different sources and ito yung mga target cells nila. So, do try to memorize them. Now, let's go to the different immune enhancers and your suppressors. So, we're done with the cytokines class, diba? So, let's start with the stimulation of the adaptive immunity. So, the stimulation of the adaptive immunity would begin with the activation of lymphocytes. And they would require two signals. So, this is very basic pa. Ulit-ulit lang tayo dito, class, in regards to the activation of lymphocyte. The first one, for a lymphocyte to be activated, whether it's a T cell or a B cell, We're talking about here the small lympho ha. For it, and even the NK cell class, pwede rin siya maging activated. Is that you have to see, they have to encounter an antigen. So two signals. The first signal is that they encounter an antigen. The second signal is that molecules that are produced during innate immune response to microbes or injured cells. So this is a two signal hypothesis. So remember that the two signals involved for the activation of lymphocytes, whether it's the small lymphocyte, T cell, B cell, or the NK cell, is that they have to be an antigen. And the second are the molecules produced during innate immune response to microbes or injured cells. This is a two signal hypothesis. So dito siya class. So the first signal, di ba, as mentioned earlier, is that it has to be an antigen. So this is your microbial antigen. They would directly attach to the receptor, to the antibody. The second one, whether the antibody or the T cell or the NK cell, the second one is molecules induced by innate response. Pwedeng yung mga complement fragments. So when they encounter these molecules from your complement fragments, co-stimulators, itong dalawang signal to na to class, they can cause lymphocyte proliferation and differentiation. So meaning class, the dami yung increase yung quantity nila, proliferation, and they would differentiate. So they would either be, if it's a T cell, it's going to be a T helper or a T cytotoxic or suppressor. If it's a B cell, here in your adaptive immune response, it could either become a plasma cell or a memory cell. So, yun yung ano. Now, cytokines in innate immune response. So, let's just have a recap. So, interleukin 12 will stimulate the differentiation class of naive CD4 positive T cell to the T helper 1 subset of effector cells. So, siya yung nagde-differentiate class between inactive CD4 positive T cell from the T helper 1 subset. Then interleukin 1, 6, and 23 stimulates the differentiation of naive CD4 positive T cell to the T helper 17 subset of the effector cell. So, pakitandaan. Then interleukin 25, interleukin 33, and TSLP class would stimulate the differentiation of uh, naive CD4 positive cells to the T helper subset of effector cells. Now, itong TSLP na to, please take note that this is a type of protein known as your stromal thymic, THYMIC, stromal lymphopoietin. So, siya yung nagdi-differentiate ng naive CD4 to the T helper subset of effector cells. Then, interleukin 15 would promote the survival of memory CD8 positive T cell. Then, interleukin 16 promotes the production of antibodies by activated B cell. Now, mechanisms that would limit the innate immune response. The first one is interleukin 10. This is a cytokine class that is produced by and would inhibit the activation. So this is an inhibitor class of macrophages and dendritic cell. It inhibits the production of various inflammatory cytokines by activated macrophage and dendritic cells, including interleukin 1, TNF, and interleukin 12. Excellent example of negative feedback regulator. Then we also have your interleukin 1 receptor antagonist or your IL-1 IRA. Functions as competitive inhibitor of interleukin 1. Major function is to act as a decoy glass that competitively inhibits interleukin 1 binding to the type 1 signaling receptor. So please take note of that. Decoy siya class. and it would competitively, meaning nakikipag-agawan siya. Nakikipag-agawan siya in inhibiting interleukin 1 by binding to type 1 signaling receptor. Then secretions of inflammatory cytokines from a variety of cell types appears to be regulated by the products of autophagy genes. Now, autophagy genes class would involve the mechanism which would impair cytokine synthesis are not yet well understood. They may regulate inflammasome activation or production of reactive oxygen species. Now, when you say autophagy class, you're basically self consuming or self killing. So this is parang starvation class. This is a type of apoptosis. So sabi dyan, mariregulate daw siya by those genes. That's how they would control it. Now, there are numerous negative regulatory signaling pathways that would block activating signals generated by pattern recognition receptors and inflammatory cytokines. Now, kapag kasi nagkaroon ng sobrang dami ng cytokines, such as yung tinatawag nating cytokine storm. Let's say nagkaroon ng cytokine storm, naka-encounter kayo ng super antigen. What happens is that there are different negative regulatory pathways. So may mga pathways yan. An example of this is your SOCS proteins or your suppressor of cytokine signaling proteins. Inhibitors of the JAK-STAT signaling pathways linked to cytokine receptor. Then SOX proteins would limit response of this cell to the exogenous cytokines such as type 1 interferon. Then SHP-1 negatively regulates pro-inflammatory responses of cell to TLR signaling. Activating and inhibitory receptors of NK cells. So NK cells would distinguish infected and stressed cells from healthy cells. And NK cell function is regulated by a balance between signals class that are generated from activating receptors and inhibitory receptors. So this is the NK cell activation overview. As mentioned earlier class, ang NK cell nyo, kaya niyang makapag-recognize ng healthy from an infected, damaged, or tumor cell. Now, kapag naka-encounter siya ng healthy cell, hindi ma-activate yung NK cell niya. But if it encounters an infected, a damaged, or a tumor cell, it would become activated, killing the abnormal cell. So if you still remember, di ba, nag-a-attach siya dun sa antibodies, tapos sisirain niya rin class yung cell, yung antigen. But whenever there would be too much NK cell, nagkaroon ng inhibitory receptor. Let's say nagkaroon ka ng inhibitory receptor, here you have your normal cell. Now, your normal cell using the MHC class 1 would introduce this to the inhibitory receptor. Now, pag nangyari yan, the inhibitory receptor will prevent the NK cell from activating. And when that happens, class, no cell killing will occur. And this is due to the presence of the MHC class 1. MHC class 1 basically stopped or prevented the activation of your NK cell. Another one is the inhibitory receptor not engaged. So this one is wherein the virus would inhibit class 1 MHC expression. So instead of the class 1 MHC na mag-inhibit, this time yung virus naman. And when that happens, class, the NK cell or the signaling pathways will not come into action, thus preventing the killing of infected cell. So this is the fate of activated naive B cell. So parang nag-discuss lang tayo class ng life cycle niya. Yung cycle ng from young, from immature to mature. So here you would have your naive B cells. Now when your naive or inactivated B cells bind antigen, it would start to form they would bind to the membrane-bound antibody. So if naalala nyo, di ba, ito yung B cell nyo. Sa surface ng B cell nyo dyan, merong antibody. So the antigens would attach here, and this would followed by endocytosis of the antibody-antigen complex. So pag sinabi mong endocytosis, itong antigen class, papasok yan dyan sa B cell. Kakainin niya. And it would be destroyed, slowly destroyed, and it would be presented as an antigen fragment. So magiging molecules na siya dyan as a class 2 MHC molecule. So ipepresent siya using the class 2 MHC. Now, kapag na-present na siya, it will now be presented class 2 and activated helper T cell. Si helper T cell nyo, kapag na-present na yung molecules ng antigen na yon, it will now start secreting cytokines, which would lead to the clonal expansion of the naive B cell. So, pag sabi mong clonal expansion, dadami siya. And sa pagdami niyan, it could either isotype switch. So, pwede na siyang maging IgM, IgG, and so on. Depende sa ano. And they would have affinity maturation. So, they would have only a specific target. Or another possibility, pwede lang siya maging IgM secreting plasma cell. And this is seen class in primary infections. So, meaning first time niyang na-encounter. So, pagiging IgM, pag first time niyang na-encounter yung IgM, pwede siyang maging IgM secreting plasma. Pero kung na-encounter na niya or this is a re-encounter, let's say second time mo nang nagkaroon ng sakit na yan, there will be isotype switching. So, pwede na siya maging IgG and so on. Now, when there is isotype switching, this is followed by further clonal expansion. So, dadami na sila na dadami. Then, they could either become IgG-secreting plasma cells that die after 3 to 4 days, or they become further memory B cells that would circulate into the immunosurveillance. So, that's what happens to your naive B cell. Now, how do you turn the B cell response class, or how do you stop it? So this would occur class when IgG antibodies bind to the Fc gamma receptor present on the B cells and antigen crosslinks that IgG antibody with the membrane-bound immunoglobulin. So ganito yung class. Kunwari ito yung B cell nyo. Yung B cell nyo class would have the Fc gamma receptor. So kunwari ito yung Fc gamma receptor. Now what happens is that when your yung IgG mo mag-attach na dyan, it would now form crosslinks that would bind to the membrane immunoglobulin. So, nakabound na yan dyan. Now, the cross-linking of these antibody molecules will trigger a signaling pathway that would transmit a negative signal to the B cell so that it will not differentiate into antibody-secreting plasma cell. So kapag nakabind na dyan class yung IgG dun sa Fc gamma receptor ng B cell, magsisend na yan ng signal na tama na, stop ka na sa pag-produce or pag-differentiate into antibody-secreting plasma cell. So remember this class, the way you would turn off B cell response is when your IgG antibodies bind to the Fc gamma receptor sa B cell. Now, signals required for activation of CD4 positive T cell or yung helper T cell. So, naive CD4 positive T cells are required, or these are also termed as your T helper precursor cells. Now, signals required is that first is a T cell with an antigen presenting cell that is displaying an antigen fragment MHC class 2 complex. T cell co-stimulatory molecules with their counter receptors on the antigen presenting cells because this generate important intracellular signals such as your CD28 on T cells with B7 on the APC. Now, CD4 molecules with class 2 MHC molecules at the site distinct from those sites recognized by the T cell receptor. CD4 is a co-receptor that is required for optimal activation of T cell because it participates in intracellular signaling event. addition molecules present on the T cell with their counter molecules on antigen presenting cells to stabilize the T cell APC conjugate. Differentiation of THP cells leads predominantly to a helper Th1 or Th2 T helper 2 cell phenotype. So ito siya class. Activated T helper precursor cells will clonally expand. They would differentiate into your inactive or your naive T helper intermediate cell that will further differentiate into T helper 1 or T helper 2. T helper 1 will secrete type 1 cytokines for immune response involving macrophages, T cells, and NK cells. Then your T helper 2's main role is to secrete type 2 cytokines are needed mainly for immune response involving B cells. Now let's talk about the fate of activated CD4 positive T cells. So most activated cells die due to programmed cell death or apoptosis. Some will differentiate to memory cells that will circulate into immunosurveillance. So it's either they die through apoptosis class or maging memory cells sila. Okay, let's continue with this one. So we also have the CD8 positive T cell antigen encounter. So when naive CD8 positive T cells would leave the thymus, they are known as your pre-cytotoxic T cell. So paglabas nila ng thymus class, yung mga CD8 positive cell, ang tawag sa kanila is the PCTL or the pre-cytotoxic T lymphocyte. Now they would circulate throughout the body on alert class for infected cell, meaning class they would tend to look or they would search. Now in contrast with CD4 positive T cells, the initial encounter of a PCTL with an infected cell may occur anywhere in the body and not just in a secondary lymphoid tissue. So class sa PCTL nyo, nangyayari siya anywhere. Pero yung CD4 nyo, it would just, activation would just occur in the secondary lymphoid organ, sa spleen, sa lymph nodes. Now, activation and differentiation of PCTL to CTL. So this would require the interaction of class 1 MHC on the infected cell with CD8. This is required for optimal activation of the pre-cytotoxic T lymphocyte. CD8 participates in intracellular signaling events. Activated PCTL are induced to express high affinity interleukin 2 receptors that would bind interleukin 2. Interaction of interleukin 2 with the high affinity interleukin 2 receptor leads to clonal in fact in expansion of the PCTL. So please take note class si interleukin 2 ang the one responsible for its clonal expansion. Now, what is the fate of your CD8 positive T cell? So, not all of the CTL that constitutes a clone, however, are required for host defense. Once the infected cell has been destroyed, most activated cells will die due to programmed cell death. Some will differentiate to memory cells that would circulate in immunosurveillance. So, halimbawa, class, meron siyang antigen na detect. This would lead to clonal expansion. Dadami siya. And these clones would only target that specific antigen. Kaya siya tinawag na clonal. Now, when that happens, class, let's say nasira ng antigen, most of them will die through apoptosis. And some class will differentiate to memory cells, becoming responsible in immunosurveillance. Now, memory T cells class, memory CD4 T cells, and CD8 positive cytotoxic cells would recirculate to detect the same antigen fragment MHC complex triggered by their initial differentiation. Memory cell formation would receive a rapid response. Memory CD4 positive T cells can be activated at the site of infection and not solely in the secondary lymphoid tissue. CD8 positive T cells can be activated at the site of infection, irrespective class of whether this is an initial response or a subsequent encounter. So please take note of that class. Kaya siyang maging activated whether it is an initial or a secondary or subsequent encounter. Now let's go to the role of your T helper in the immune response. In cell-mediated immune responses class against phagocytosed microbes, T cells would specifically recognize microbial antigens, but phagocytes would actually destroy the pathogen. Ingestion and elimination of microbes by phagocyte is also a major reaction of your innate immunity. But T cells would greatly enhance this function by phagocytes. Inflammation consisting of leukocyte recruitment and activation would accompany many of the reactions of CD4 positive T lymphocyte and may damage normal tissue. Now, what are the different subsets of CD4 positive effector cells, yung T helper? Ang alam natin class or was discussed previously is yung T helper 1, T helper 2. But this time, there is another subset, yung T helper 17. So the signature cytokines produced by the major CD4 positive T cell subsets are enumerated. So si T helper 1 class would secrete interferon gamma. Si T helper 2 would secrete the cytokines interleukin 4, 5, and 13. Si T helper 17 would secrete interleukin 17 and 22. So please take note of the cytokines secreted by the different CD4 positive cells or your T helper. Pakitandaan class ha, kapag tinawag mong CD4 positive, we're talking about here the T helper. Kapag CD8 positive, si T cytotoxic yan. So huwag niyong pagkakabalik na rin or now, these are the different targets, immune reaction, host defense. So, sige, let's tackle them one by one. Si T helper 1 would secrete the interferon gamma. Ang target cell niya class is the macrophage. And what is its effect? It causes your macrophage to become activated, responsible in the defense against intracellular pathogen. Role in disease specifically involved class in autoimmunity and chronic inflammation. Then we have your T helper 2 as mentioned previously si T helper 2 class involved yan sa parasitic at allergic. It secretes 4, 5, 13. Interleukins 4, 5, 13. Ang target cell is eosinophil. It causes the activation of eosinophil, mast cell, and also in an alternative way, macrophage activation. Plays host defense against helminths. Role in disease specifically involved in allergies. Then we have your T helper 17, secretes interleukin 17 and 22. Target cell is neutrophil, responsible for recruitment and activation. Host defense against extracellular bacteria and fungi. Role in autoimmunity and inflammation. Then we have your function T helper cell, secretes interleukin 21 and interferon gamma or interleukin 4. The target cell is your B cell. Major immune reaction is antibody production. Host defense against extracellular pathogen. Role in disease, autoimmunity. Specifically auto antibodies, which we will tackle class in module 10. So this is the role of your T helper cells in a cytotoxic T lymphocyte. So CD4 positive T helper cells would produce cytokines that stimulate CTL differentiation. So upon secretion class, ito yung helper nyo. Si CD4 helper nyo, when it encounters an APC, magsisecrete yan ng cytokines. Si cytokines nyo class will help the cytotoxic or the CTL, so the cytotoxic lymphocyte to differentiate into either activated CTL or a memory CD8 positive T cell. Another one is when your CD4 positive helper T cell would enhance the ability of APCs to stimulate differentiation. So instead of directly helping them in differentiation, it uses the APCs. So gagawin ni CD4 positive, it would assist the the antigen presenting cell to stimulate CTL differentiation. So dalawang ways class siya pwedeng makahelp sa CTLs nyo in differentiation. It could directly stimulate CTL or it could use APCs to stimulate CTL differentiation. Now let's go to the last topic class, yung tinatawag nating apoptosis, or in general term the program cell. So a normal homeostatic mechanism is necessary for maintaining uh, normal cell number class. So your body on its own would tend to kill cells class, and that is for the purpose of balance or homeostasis. Now this would occur class through a series of biochemical reactions within a cell that would lead to chromatin condensation, DNA fragmentation, cell shrinkage, and membrane bleeding. Now, there are two pathways: the mitochondrial or the intrinsic pathway, or the death receptor or the extrinsic pathway. Okay, so to explain this, new slide. Okay, siguro let's start first with the mitochondrial pathway or the intrinsic pathway. Now, the thing you need to remember about the mitochondrial or the intrinsic pathway class is that regulated. They are regulated by BCL, BCL2 proteins. Now, ano ba itong mga BCL proteins na to? That these are the ones that maintain or basically control the apoptosis through the intrinsic pathway. Now, first you have to understand, class, that the main organelle here is involved is your mitochondria. Now, inside your mitochondria class is that you would find different substances, and one of those is the substance cytochrome cytochrome C, which is responsible, class. Please take note na si cytochrome C would prevent prevent apoptosis. It would prevent apoptosis by staying inside the mitochondria. The moment na lumabas yan, class, the moment na lumabas siya ng mitochondria, it will trigger this intrinsic pathway. Now, another thing you also need to understand, class, is that inside the cytoplasm, sa cytoplasm ng cell, is where you would find a substance known as your APAP1. APAP would stand for apoptosis activating factor 1. Let me repeat. AFAP stands for apoptosis activating factor 1, which, class, must never come into contact with cytochrome C. Let me repeat, class. Your mitochondrial pathway or your intrinsic pathway is regulated by BCL2 proteins. Understand also that inside the mitochondria, there is the substance cytochrome C, which prevents apoptosis by staying inside the mitochondria. And understand also that inside the cytoplasm, there is a substance known as your APAF1 or your apoptosis activating 1, which must never come into contact with, with cytochrome C. Now, this pathway, class, please take note, is initiated. Initiated siya This is initiated class when cytoplasmic proteins when cytoplasmic proteins belonging to the BH3 family belonging to the BH3 family belonging to the BH3 family binds to the proteins BAX and BAK. When this happens, class, this will cause increase mitochondrial permeability. And again, 'di ba, kapag permeable ka, you can easily pass through. And due to this mitochondrial permeability class, si cytochrome C nyo will go into the cytoplasm where it would mix with your APAF1. And when that happens, class, it causes or it activates an enzyme known as it activates the enzyme caspase 9. It activates caspase 9 from procaspase. Naalala nyo sa hematon nyo, di ba meron tayong mga tinatawag na zymogens. Kapag sinabi mong zymogens, mga inactivated form. Di ba pag naging activated yan, bawa yung factor 12 nyo, pag nilagyan nyo ng A, magiging activated. So, ang case natin dito, yung procaspase, that is the inactivated. So, ang gagawin, pag nag-combine yung cytochrome C sa APAF1, it would cause your procaspase to become caspase 9. And itong caspase 9 na to will lead to DNA fragmentation, DNA damage, and hanggang sa masira na yung buong cell. So, para mas makita nyo ito sa class. So, this is the mitochondrial intrinsic pathway. So, there are proteins, yung BH3, yung BAC, saka yung BAC. Now, inside the mitochondria class, ito yung mitochondria, there is the cytochrome C. Itong nakikita nyo na to. Yan yung cytochrome C na yan. That must never, let me repeat, that must never go into the cytoplasm. Kasi once it goes into the cytoplasm class, it will cause the binding with your APAF, and that would cause your procaspase to become activated, to become caspase 9, leading to destruction of the cell. Mangyayari lang yan, lalabas lang yan si cytochrome C when these BH3 proteins bind to the box and the back. So, try to understand this, class. I'll give you the recording. Pangit ng sulat ni sir. Sorry ha. So, yun. So, I hope that was explainable yung mitochondrial or yung intrinsic pathway. Let's now go to the last pathway or yung tinatawag nating death receptor or yung intrinsic pathway. Teka na, try ko nga kung pwede magsulat na lang ng teka kung pwede na lang magsulat ng wala ka na bang ikakaganda ng sulat, sir? Wait. Hindi naman pwede. Okay. Sige, lakihan ko na lang para hindi ano. Okay, this one. Okay, please take note, class, that the second pathway, your extrinsic pathway, also known as the death receptor pathway, Now, this pathway naman class, instead of involving other proteins, this would involve the so-called fast receptor. Kasi fast receptor nyo, para mas maintindihan, si fast receptor nyo class is found on the surface. This is found on the surface of the cell. Now, on the side of it is where you could find the so-called fast associated death domain, also known as the FAD, which would bind to inactive pro space 8. And again, di ba nga itong mga procaspase na to, this is another type of inactivated enzyme uh, that can cause apoptosis. Now, what happens in this case, class, is that in a healthy cell, inhibitors are present that prevent fast receptor from activating pro-caspase 8. So in a normal cell class, nagre-release yan na yung ng inhibitors that would prevent yung fast receptor from activating pro-cas pro-caspase 8. Now, this pathway class would become activated when it becomes a tumor cell. When you say tumor cell, we refer to it also as cancer cells. And when this happens, one cell, a cell known as your CD8 positive cell or your cytotoxic T cell will arrive and will attach to the fast receptor using a so-called fast ligand, also known as the fast L. So, ulitin ko class ha, your extrinsic pathway or death receptor pathway involves the so-called fast receptor, which is found on the surface of the cell. And on its side class is your so-called fast associated death domain, which would bind to inactivate procaspase 8. Now, in a healthy cell, parang ganito yan. Kunwari ito yung cell nyo. Dito sa surface niya, nandito yung fast receptor. Dito sa gilid, nandito yung FAD. Yan, yung dalawang yan. FAD yan class, yung fast associated death domain. Tapos dito, sa gitna, naka-attach dyan yung pro-caspase 8. Yung pro-caspase 8. Now, dito class sa gitnang part na to, may mga inhibitor proteins dyan. Now, it would prevent itong inhibitor proteins na to, it would prevent the fast receptor from activating procaspase 8. Now, the only way for this pathway to become activated class is when it would encounter a tumor cell or when the cell becomes a tumor cell or a cancer cell. Now, when this happens class, a cell known as your CD8 positive cell or your cytotoxic T cell will arrive and will attach to the fast receptor using the fast ligand. And when this interaction occurs, class, pag nangyari itong interaction na to, when this interaction occurs, changes in fast receptor leads to FAD trigger activation of pro-caspase 8. So yung FAD class, hindi niya na may stop yung pag-activate ng procaspase 8. And this will now lead to become caspase 8, which would initiate caspase class that would activate BH3 protein, which, as you all know, in the intrinsic pathway, yung BH3 protein nyo will also bind to box and back, leading to the apoptosis via mitochondrial pathway. So, nagkikita pa rin sila, class. It's similar with your coagulation pathway. Di ba yung extrinsic at intrinsic pathway yung nagkikita sa common pathway. Itong extrinsic pathway ng death receptor nyo, it still leads back to the mitochondrial pathway. So, para mas maintindihan nyo, let's use this picture. Okay. So dito tayo sa death receptor or intrinsic pathway. Now this, as mentioned, this will activate class when the cell would become a tumor or cancer cell. So as mentioned, sa surface niyang cell natin merong FAS. Ngayon dadating si T cell class, si T cytotoxic or yung CD8 positive. It will cause the attachment of your FAS ligand. And pag nangyari yon, again, yung mga FADD dito sa gilid will no longer be able to inactivate procaspase. And it will cause the activation of your caspase 8. Si caspase 8 nyo ngayon, class, will now cause the activation of BH3 proteins. And that would also lead to the apoptosis via the mitochondrial pathway. So that ends the module 8. So these are your references. So I hope it was clear. Thank you.